De novo assembly of genome. De novo assembly uses DNA sequencing reads to build a genome sequence from scratch. So basically it's used when a reference genome for the organism of interest is not available. How it works? So in brief, it starts with overlap detection. Assemblers identify overlapping regions between sequencing reads. Next step, contig construction. Then, overlapping reads are joined to form contigs, which are continuous stretches of DNA sequence. Scaffolding. Contigs are connected into larger sequences, that is, scaffolds, using information from paired end reads or other data. Finally, gap closure. Then, gaps between contigs or scaffolds may be filled using additional sequencing data or other methods. This was all. Thanks for watching.